Hello everyone, my name is John Ohiolei from Lanzhou Veterinary Research Institute, Chinese Academy of Agricultural Sciences. And I'm most delighted and happy to participate in this year's World Conference on Sheep. The topic before me is on the status of cystic echinococcosis and cysticercosis in sheep in the Tibetan Plateau. To get us started quickly, I would like us to look briefly at the geographical coverage of the Tibetan Plateau and some important information. Then we'll look at the biology and the life cycle of the causative agents of the infections, then the prevalence and the genetic variation, diversity, as well as the implication for control. The Tibetan Plateau is a very large mountainous region in China. It has an average elevation of 4,000 meters and covers several provinces and autonomous regions. Scientists believe it was created by a massive geological event that occurred millions of years ago. And this plateau is known for its harsh climatic conditions and peculiar biodiversity, as a number of new species have been uncovered in the plateau in recent years. Animals inhabiting this region are highly adapted to this harsh environment and meteorological nature. In the plateau, the inhabitants are mostly pastorals, and there is this limited social amenities available in the region. So far, the cestodes found in this region are mostly maintained through a wildlife domestic interface, as well as the feeding relationship between the animals in the region and also the lifestyle of the inhabitants. Studies have shown that cystic echinococcosis and cysticercosis are also co-endemic in the plateau. Cystic echinococcosis and cysticercosis are both tapeworm infection. Why cystic echinococcosis is caused by Echinococcus species, a rather complex species with different genotypes. Cysticercosis, on the other hand, is caused by Taenia hydatidina. Now, the adult worms live in the small intestine of the definitive host, which are mostly carnates, and these hosts release their eggs from this adult tapeworm through their feces into the environment. These eggs are thus infective to ruminants, and in the case of echinococcus, it is infective to humans as well, so that makes it zoonotic. Now, the eggs are well adapted to survive in the environment for a very long time and are mostly better surviving in moist conditions. The intermediate host, in this case the sheep, becomes incidentally affected while grazing or foraging. The eggs hatched in the small intestine to release the oncosphere, which penetrates the gut wall and are carried in the circulatory system to various organs, where they form metacestodes, which is the cyst. This is common, a common pattern of development both for echinococcus and Taenia hydatidina, virtually for most tapeworms. Why the Taenia hydatidina utilizes the dog mostly, echinococcus on the other hand utilizes the several carnates, dogs, wolves, Tibetan foxes, and so on. The picture shows what an infected lungs or liver looks like in the case of echinococcosis. A fluid-filled sac that grows slowly and are usually asymptomatic, until they are large enough to put pressure in the adjoining tissues. So they can cause pain when they become so large. Otherwise, you will not even notice any symptoms. But generally, so far, symptoms that have been reported occasionally in sheep include hepatic disorders with ascites and jaundice, as well as bronchopneumonia, heart failure, poor growth, weakness, and lameness. The detection of these tapeworm for echinococcus is usually during slaughter and most times by serology. Globally, the financial loss as a result of this infection, cystic echinococcosis alone, amounts to over 3 billion US dollar annually, and this is basically due to treatments in humans that are infected and also losses in livestock value. The prevalence of echinococcus species, which is a causative agent for cystic echinococcosis, in northwestern China, where it is very endemic, which is the Tibetan Plateau, was previously very high, even as high as 82%. However, since the National Control Program started in 2006 across this Tibetan region, which is the endemic region, the infection has reportedly dropped. And in 2019, there was a report showing the prevalence of cystic echinococcosis among sheep in the Tibet Autonomous Region to have dropped up to as low as 11%. Now you can see from 82% down to 11%. Most recently, in one of our study, that was conducted also in the Tibetan Plateau, in Xinjiang Autonomous Region, we found a prevalence of 2.4% on the average. This was from over about 5 cities or counties. That is quite a drop, and this will say thanks to the control efforts so far made. Now, the genetic variation and diversity of echinococcus species in sheep in this plateau, in this report, we analyzed over 182 isolates from Tibet Autonomous Region and Xinjiang Autonomous Region. And so far, about 99% of the infection in sheep is mainly caused by the G1 genotype. If you recall, I told you echinococcus species is a cryptic species made up of several species and genotypes. So in sheep, the G1 genotype is mainly responsible in the Tibetan Plateau. Then the 1% is caused by, the rest 1% is caused either by the G3 genotype or the G6 genotype. Now, from the median joining network you see on the screen, that was constructed based on the NAT5 gene, you would notice that there is a major haplotype, which was also found both in Tibet and in Xinjiang. And the population indices showed that the population structure or demographics of this particular species in sheep is expanding. So it means that there is no, event that has disturbed the evolution of this particular parasite tapeworm over their their history. Now, you would also notice from the network that there are few, genetic mutations. So that indicates a very low genetic variation among these species in the Tibetan Plateau. For Taenia hydatidina, which is responsible for cysticercosis, cysts are usually found in the omentum, the mesentery, and the peritoneum, and also the liver surfaces. Less frequently, you will find them in other regions like the pleura and pericardium, and occasionally in the lungs and kidneys. One of the signs or the clinical presentation of this infection is depression and weakness. And this is secondary to liver damage. And mortality is usually high in lambs. And you can see on the screen on the slide, you would notice the liver damage that is caused by this infection, and that you can see in a lamb that was infected by this tapeworm. The detection is also similar to echinococcosis, because you would not, easily uh, detect it except during slaughter, and most times serology. The financial losses resulting from this infection is also very high, from 65,000 to 350,000 US dollars. That is even, estimated per slaughterhouse. So you can imagine the total cost for different slaughterhouses where the infection is common or endemic. The prevalence of this particular tapeworm is poorly documented in China. And this could be due to the fact that this particular tapeworm is not a zoonotic parasite, so it may not be of serious importance. However, it remains a very significant issue for livestock production. To date, most work were reported, outside the plateau, mainly in the Inner Mongolia region, and that was around the 1990s. And recently, one of the studies from the Tibet Autonomous Region, which is Tibetan Plateau, reported the prevalence of up to 52%, which is quite high. Although control effort made for the management of echinococcosis, cystic echinococcosis, could have also, impacted on the prevalence of cysticercosis, because they utilize a similar transmission pattern. What we did recently, we tried to investigate the genetic variation of Taenia hydatidigena, the causative agent of cysticercosis, in the Tibetan Plateau. And interestingly, from the result, we found two major haplogroups, two clusters of Taenia hydatidigena responsible for the infection. And we designated them as haplogroup A and B. Now, both haplogroups were also confirmed, because in that previous study we used just a number of genes, just 2 or 3 genes to check, and we were able to confirm the occurrence or the existence of these two haplogroups based on the complete mitochondrial genome sequences. And interestingly, we found over 160 mutation points or steps separating the group A from the group B. And this was found in sheep and also in the definitive host, the dogs. Now, the nucleotide changes were both, transition and transverse changes. And the population diversity estimated for both haplogroups demonstrated a significant deviation from neutrality for group A, unlike group B, where the population, the deviation from neutrality was not significant. The a pairwise comparison between the members of group A and group B showed that the group A were genetically different from group B. Now, the study shows or confirms that the species Taenia hydatidigena responsible for cysticercosis in China may be made up of several, mitochondria, several different strains based on their mitochondrial genome. And this is very important. And the study also confirms differences in their population indices. Why one is undergoing a serious population expansion, the other group did not show much deviation. Now, we believe that exploring the genetic variation of Taenia hydatigena populations across the Tibetan Plateau and across the globe in sheep may also provide a better understanding of their diversity. And we so far from this report, the haplogroup A was common in sheep, unlike the haplogroup B. So it could also represent a pattern observed in echinococcosis, that shows, cryptic, composition. What are the implications of this genetic diversity? For echinococcus, like I mentioned before, the understanding of the genetic variation and diversity among the species among the group reveal that different strains or genotype are specific to different animals. And for sheep, the G1 genotype has been found to be highly specific and to sheep. So that means the sheep is susceptible more to the G1 genotype. Now, in the case of Taenia hydatigena, our recent studies have confirmed that there are two major groups in Taenia hydatigena responsible for cysticercosis. And one of the group A, is was detected more in sheep. So understanding of this genetic variation could, for the case of echinococcosis, has so far assisted in control by helping to target specific intermediate hosts responsible for infection in a given community. So if you want to control or manage the disease, the infection in a sheep endemic region, then you have to target sheep. So that is one of the importance of this genetic variation. And this understanding will inform on the best approach to control. And also, during the production of vaccine and drugs, we have to also consider the influence of these genetic differences in how these particular hosts would respond to the drug being designed. And these are some of the very important and implications for control based on the genetic variation. The phylogeography and species distribution is also important. For example, we have seen that the haplogroup A for Taenia hydatigena is common in sheep and in China. And a preliminary study we conducted between Nigeria, China, and Sudan revealed that the haplogroup B was only found in China. So if we understand some of these, species distribution, we can be able to design, the best approach to control some of these infections. Know that some of these genetic infection variation tend to influence the response of certain drugs and vaccine, and also helps to know which host to consider when trying to break the transmission cycle. Finally, I would like to thank members and staffs of the State Key Laboratory of Veterinary Ethological Biology of Lanzhou Veterinary Research Institute, Chinese Academy of Agricultural Sciences. And I would also like to thank our funding bodies, Central Public Interest Scientific Institution, Research Fund, and also the National Key Basic Research Program. I think this is pretty much what I have to share with us today, and I do hope we enjoy the rest of the meetings. I'll sit back and wait for further questions if there is any. So thank you very much for your time.